All right, I'd like to thank the organizers for this wonderful conference and for inviting me. Actually, I have this already. So the title of the session is Descent with Modification. And of course, this was Darwin's fundamental breakthrough in understanding the organization of life. Here, of course, are the finches from the Galapagos Islands, and they all share a common ancestor, but they've diverged in both their form and in their behavior. So Darwin realized that we can understand the similarities due to homology because of common descent. And this has important implications for medical research. Rodents are often used as stand-ins for humans because of our similarities. Now, I'm from Georgia, so I can say things about similarities with rats. But of course, rodents themselves exhibit a diversity of lifestyles and behaviors, from beavers to naked mole rats to flying squirrels. And I'm borrowing a slide from Leah Krubitzer here, showing that even within the rodent clade, though, analogous behaviors evolved repeatedly and independently. So even among this diversity, there are many examples of similar behaviors evolving independently. So here the yellow lines represent diurnal behavior, which evolved independently, for example, in the tree squirrel and in Gundis. But gliding, we mentioned flying squirrels, but that also evolved independently in scaly-tailed flying squirrels, which are not true squirrels. Bipedal hopping evolved independently multiple times, for example, in the spring hare and in the kangaroo rats. And subterranean lifestyles also evolved independently multiple times, in this example, in the pocket gopher and in prairie dogs. Even eusociality evolved twice within the lineage of the naked mole rats, once in African mole rats, sorry. The naked mole rat and the Damaraland mole rat each evolved eusociality independently. Eusociality, of course, also involved in insects and in crustacea. Within bees alone, it evolved independently twice. So what does this mean about the control of these behaviors by the nervous system? Well, vocal learning evolved independently twice and maybe 3 times in hummingbirds and in parrots and songbirds. The brain areas that are involved in vocal learning seem to be homologous. They show up with immediate early gene expression during tasks involved in vocal learning. Skilled hand, I'm borrowing from Leah's work again, skilled hand use evolved independently, possibly 3 times in primates, and each time it's associated with the presence of parietal area 2, this pink area in the brain. So these homologous brain areas are involved in independent evolution of behavior, and there's been a tremendous diversity in the types of behaviors. So what neural mechanisms underlie both behavioral divergence and homoplasia within a clade? Well, let's take, for example, imagine that there are these different behaviors in this clade of organisms. There's behavior 1, 2, 3, and an independently evolved behavior 2. One possibility is that the underlying neural circuits equal the behavior. So wherever you see a different behavior, there's a different neural circuit, and behavior 2A is the same neural circuit as behavior 2. It's re-evolved the same. These two have converged on the same behavioral circuit. Another possibility is that there are multifunctional circuits, that it's the same neural circuitry, but it's producing different behaviors for these different conditions. And this is a proposition that's championed by George Lauder. So I'm going to address these questions in this group of organisms. These are sea slugs, and you're probably not familiar with them, or some of them. They all belong to a monophyletic clade called Nudipleura. Pleurbranchia, which is shown down here, belongs to a different clade than the other 3, which are nudibranchs, so the other 5. We can divide up, this is just showing additional representative genera just to fill out the tree, but there are actually thousands of nudibranchs around the world. These 5 nudibranchs form a monophyletic clade called Cladobranchia. they can be subdivided into two clades. So there's aelids, which Hermesenda belongs to, and then there's the dendronotids, which Dendronotis, Melobe, and Tritonia all belong to. There's a third clade down here, the dorids, which I'll just mention briefly. These animals conveniently express two different types of swimming behaviors. There's swimming behaviors which involve dorsal ventral body flexion, and then the swimming behaviors that involve the left-right body flexion. And then there are many animals that do not swim at all, and they're distributed throughout the tree. So Tritonia and Pluribranchia are both dorsal ventral swimmers, as are two of the dorids. This is an example of this dorsal ventral body flexion, showing Tritonia escaping from a sea star. So this is in real time. Here's the escape response. It involves a ventral body flexion followed by a dorsal body flexion. Now, it is generous to call that swimming. It's really flailing, but it does have specialized adaptations. The mouth and the tail both flared out to produce these paddle-like structures, and the animal does this rhythmically for about a minute, gets it up into the water, got it away from the starfish. I mean, it was a 6-second delay in the latency to the escape, but it's escaping from a starfish, for God's sake. Here's another Tritonia chomping down on a sea pen. And then at the end, the animal ends with a dorsal body flexion, and it then lands back on the substrate and it begins to crawl. These animals crawl with mucociliary mechanisms, so they have cilia on the bottom of their foot, and they glide along on mucus at about this rate. So you can see why swimming is really important to them, because the starfish really can go a bit faster than that. Okay, so Tritonia and Pluribranchia swim with this dorsal ventral body flexion. Melobe, Dendronotis, and Hermesenda all use this left-right flexion. And let me show you. Oh, and these other species also use left-right body flexions, including one of the dorids. So here's a movie that I got off of YouTube showing Melobe flexing from side to side. It's flattened in the sagittal plane. Its mouth is up here. Its foot is this, it's hard to see in this light, but there's a white stripe. That's its foot that's been flattened in that direction. And it's going back and forth from left to right. Okay, and it'll keep going. So one of the differences between Tritonia and Melobe is that Tritonia swims for about a minute, and Melobe will just continue to swim until it's found some more substrate to land on. Here's Dendronotis iris, which is called the giant nudibranch. It also swims in the same way. It's flattened in the sagittal plane. It has this white stripe as its foot, and its head is up here, its tail is down there, and it's basically doing this from side to side. Oh boy, that was just telecast, wasn't it? All right. Here in red are the non-swimmers. So Toquina tetraquetra doesn't swim, and it's the most closely related species on this tree to Tritonia. They're in the same family. And there are many other sea slugs that I'm not showing you here that don't swim, but notably there are slugs in every single monophyletic clade that don't swim. So if we try to envision what the most parsimonious explanation is for this distribution of behaviors, we can say that this dorsal ventral swim evolved independently 3 times, once in Pluribranchia, I'm not showing you the whole clade, once in the Dendronotids, and then once in the dorids and once in the Dendronotids. Whereas lateral flexion swimming evolved at minimum twice, once in the dorids and once in the nudibranchs, in the cladibranchs. And then swimming was lost several times, according to this phylogeny. So let's talk about what the possible neural mechanisms are underlying these swimming behaviors. So we're first going to compare the differences between Melobe and Tritonia. The advantage of working on these sea slugs is that they have individually identifiable neurons that we can go in and record from for hours at a time and then work out the circuit diagrams. So here is the circuit diagram for the swim central pattern generator. I'll use the term CPG too sometimes. For swimming in Melleby. So there's a single neuron called swim internuron 1 on the left side and on the right side. And there's another single neuron called swim internuron 2 on the left side and the right side. Which the lines with circles represent inhibitory synapses, and the resistor symbols represent electrical coupling. So what you can see is that there's strong electrical coupling ipsilaterally and mutual inhibition contralaterally. And this creates a rhythmic pattern of discharges where the left swim interneuron 2 fires and in alternation with the right swim interneuron 2. So you get left, right swim movement. So you can read the behavior right out of the pattern of discharges from these neurons. In tritonia, here are the neurons that are involved. They're called the dorsal swim interneuron, the ventral swim interneuron, and interneuron C2. The connectivity between these neurons differs. It's much more complex in its circuitry, and the electrical coupling is primarily contralateral. Each neuron is electrically coupled to its contralateral counterpart. So during the swim motor pattern, the dorsal swim interneurons, the left and the right dorsal swim interneurons fire in synchrony because the left and the right side are doing the same thing. And C2 is also doing the same thing left and right. And the ventral swim interneuron is firing in the breaks here. So here you get dorsal, ventral, dorsal, ventral, dorsal, ventral. And that's, again, the behavior that you can read out of the recordings of these neurons. And I should point out that these recordings were taken from isolated brains sitting in a dish, but you can make the same recordings in an animal that's suspended from the skin and the animal is free to move around. So we can record what are called fictive motor patterns in these cases, as well as the motor pattern underlying an actual behavior. So this is what the brains of these animals look like. They have only 10,000 neurons in their heads, and each of these circles represents the cell body of a neuron. They're actually, you can see the cell bodies under a microscope. So swim interneuron 1 and swim interneuron 2 are located here, 1 and 2, whereas C2, DSI, and VSI are located here in tritonia. These are physically different neurons. We can look at them and say that these are not the same neurons. They don't have the same morphology. They're located in different places. They're not homologous neurons. So these different behaviors are controlled by different neural circuits. However, homologs of these we've identified homologs of these tritonia interneurons in Melleby. And I want to show you that here. So here's the tritonia brain. If we blow it up, I want to point out the scale bar. So this is 200 microns. So this neuron is about 1/4 of a millimeter in diameter. How big is a Drosophila brain again? Could probably fit somewhere in there. This white cell is C2. So you can just open up a tritonia brain, every single brain, and find that white cell. And here's its contralateral counterpart. We can also further identify it by its axon projection pattern. If you fill it with a dye, you can trace out where it goes, and by its immunohistochemical profile. So this neuron is immunoreactive to a peptide called formurfamide and another peptide called small cardioactive peptide B. In Melleby, we can find a white neuron in about the same position, and when we fill it, it has the same axon projection pattern. And it is also formurfamide and SCPB immunoreactive. So we consider these things homologous neurons because the same criteria that we use to uniquely identify this neuron in tritonia can be applied to Melleby, and we can find a unique, individually identifiable neuron. The dorsal swim interneurons are located here, and we can visually identify them by eye in these species. But you can see them better if you do serotonin immunostaining. So here, these neurons are serotonin immunoreactive. Here's that giant cell that I showed you a minute ago. It's also serotonin immunoreactive, but it's involved with feeding. I should mention these neurons are found in every single gastropod that's ever been examined. So the DSIs in tritonia are serotonin immunoreactive, and there are homologs in Melleby that are also serotonin immunoreactive. They also share other properties of the DSIs. They have the same axon projection pattern, etc. In Tritonia, as I showed you a second ago, the DSIs are rhythmically active in phase with the motor pattern because they're part of the central pattern generator for swimming. In Melody, the homologs are not rhythmically active during left-right flexion swimming. So here we have homologous neurons that are playing different roles in the production of different behaviors. In Tritonia, the DSIs are both necessary and sufficient to maintain this dorsal swim motor pattern. So here in this example, I've depolarized the DSI to cause it to fire, and that is turned on bursting in C2 and another DSI. If you bath apply serotonin to the isolated brain, you turn on swimming. If you inject the animal with serotonin, the animal starts to swim. If you inject the animal with a serotonin antagonist, the animal won't swim. If you put the serotonin antagonist on the brain, you don't get a fictive motor pattern. In contrast, in Melody, you can turn on swimming with the DSI homolog, but notice the DSI itself is not rhythmically active. But serotonin and the DSIs are not necessary for swimming. So if you put on a serotonin antagonist, you still get swimming. If you inject the animal with a serotonin antagonist, the animal can still swim. So in Melody, serotonin is sufficient, but it's not necessary. So what about in a non-swimming animal? Well, again, we can identify DSI homologs in Tokina. These neurons are not rhythmically active. Here, given a nerve shock which turns on the swim motor pattern in Tritonia, but it doesn't cause this neuron to fire bursts. However, Bill Frost and Bill Frost's lab at Rosalind Franklin Medical School, they showed that the DSIs also play a role in crawling, in their crawling behavior. They activate the neurons that turn on the cilia. And here we've shown that if you stimulate a DSI to fire action potentials, you can record monosynaptic EPSPs in pedal 5, which is a neuron that secretes a neuropeptide that turns on the cilia. We've identified a homolog of that pedal 5 in Tokina, and the DSI homolog produces monosynaptic EPSPs. So this part of the circuitry is highly conserved, and this neuron is playing, the DSI is playing a role both in swimming and in accelerating crawling, and that is present in both of these species. So the DSIs are actually found in every opistobranch that we've looked at so far. So Tritonia, Melody, Pluribranchia, Cleone, which swims with yet a different motor type of movement. It flaps these wing-like structures, and Aplysia californica, which doesn't swim at all. So each of these species has these homologous neurons. They have the basic structure of the nervous system hasn't changed, but they're doing different behaviors. So Tritonia and Melody use different neural circuits to produce different swimming behaviors, but homologous neurons are present and play different roles in these behaviors. However, homologous neurons play similar roles in other behaviors, such as crawling. So the first conclusion then is that divergent behaviors are produced by different neural circuits composed of multifunctional but homologous neurons. So that's these first three species that we've looked at. Now I want to shift attention to Pluribranchia and Tritonia, which both swim with these dorsal ventral body flexions but are the most distantly related that we'll talk about in this talk. Most likely, these animals independently evolved the swimming behavior. So here again, Pluribranchia has homologs of the DSIs. These were identified in Marina Gillette's lab at the University of Illinois. They named them AS1 to 3, but I'm just going to refer to them as DSIs just to simplify it. The Pluribranchia also has a C2 homolog. This was work done by Josh Lilbus in my lab. Here's where C2 is located. There's a white cell in the same position in Pluribranchia. We can, as I showed you before, if we fill the cell, it's vermurfimid and SCP immunoreactive, and the same thing's true in Pluribranchia. So we're very confident in saying these are homologous neurons. In Tritonia, C2 is rhythmically active during the swim motor pattern, and in DSI and C2 are both rhythmically active, and in Pluribranchia, their homologs are both rhythmically active during the fictive swim motor pattern. The phase relationships between these two neurons are also similar to what we see in Tritonia, but the periodicity is increased, and that is reflective of the fact that Pleurobrankia swims faster than Tritonia. So Raina Gillette's lab is, and Zhen Jing in particular, has worked out a neural circuit with these neurons, and it looks very similar in a lot of respects to the Tritonia neural circuit. So the DSI and C2 homologs are shown in the same colors. There are other cells, so there's an A10 neuron, which I've just lumped together here, and there's an A3 neuron, neither of which we found in Tritonia. And they haven't found a VSI homolog in Pleurobrankia, but we're currently looking for that. So these circuits have similarities, but there are some differences in them as well. And this synapse in particular has an important role, we think, in swimming, but we don't see evidence for it in Tritonia at all. As I showed you already, serotonin plays a major role in the circuit. So the DSIs are serotonergic, these AS cells, the DSI homologs are serotonergic. Does serotonin play a role in swimming in Pleurobrankia? And is this modulation even present? So to address this, my student Josh Lilvis, he recorded from C2 DSI and a follower of C2. If you stimulate C2, you can record these monosynaptic EPSPs in a follower. If you precede that with DSI, that synapse gets strengthened. And this is something that we've known for more than a decade now. So he looked at the same thing in Pleurobrankia. He found, again, that you can stimulate C2 alone, get an EPSP, stimulate C2 plus DSI, and that EPSP is enhanced. So yes, the neuromodulation is present in Pleurobrankia. But there's something different about Pleurobrankia. 80% of the animals don't swim. So this is a drag when you're trying to do experiments. That you look at the animals and you make them swim, they don't respond. You take the brain out of the animal and the brain doesn't exhibit the motor pattern either. So in the animals where swimming occurred, Josh saw this. But in animals or preps, in isolated nervous systems where the animals didn't swim, he got no modulation or very little modulation. So on average, there was more modulation in animals or in preparations where he saw swimming than in preparations where he did not. All right, so it could have been that we weren't stimulating DSI right, so what Josh did was he just applied serotonin to the nervous system, and in Tritonia this causes an increase in the strength of the C2 synapses, and in Pleurobrankia that swim you see an increase in the C2 synapses in response to serotonin, and in the ones that don't swim you see no change. So serotonin is not modulating the strength of synapses in the animals that are not swimming. Well, what if we look across the phylogeny? Hermesenda is an animal that doesn't swim like Tritonia and Pleurobrankia. What is serotonin doing to the DSI and C2 homologs there? Well, first, we've identified the DSI homologs. This is work from Jim Newcomb when he was in my lab. And now Josh has identified the C2 neurons, and once again, they're fumarfamid and SEP positive. And in response to nerve stimulation, they don't show bursting, which you would expect for an animal that doesn't swim like Tritonia. When he stimulated C2 alone, he got a nice EPSP in a follower cell. When he stimulated DSI prior to C2, there was no modulation. When he bath-applied serotonin, no modulation. So just like the Pleurobrankia that don't swim, serotonin's having no effect in these animals that don't swim. So dorsal ventral swimming probably evolved independently in Tritonia and Pleurobrankia. Homologous DSI and C2 neurons are both members of the swim central pattern generator in these species, and serotonergic neuromodulation is correlated with the presence of dorsal ventral swimming, both within Pleurobrankia and across species. So neuromodulation may actually uncover latent behavior that is present in homologous neural circuits, and this is something that we're looking to try to manipulate further. All right, so those are the three species we looked at. Now here's your tree again, and now let's look at Meloby and Dendronotus, which are the most closely related animals that swim with the side-to-side movement that we'll talk about here. As I mentioned before, here's the Meloby swim central pattern generator. This is what these neurons look like. Here's swim into neuron 1. its cell body's here. It has this interesting morphology. Here's swim into neuron 2. It projects through this commissure and has this dendritic branching, this axonal branching. Here's swim into neuron 1 and swim into neuron 2. In dendrinotus, we can identify them on morphological and neurochemical bases. And here's this neural circuit that they form. It's different than in Melody. So whereas in Melody there was a lot of cross the midline inhibition, in dendrinotus there's very little inhibition across the midline. Only the swim into neuron 2s are mutually inhibitory across the midline. In Melody there was strong ipsilateral electrocoupling, but in dendrinotus there's strong contralateral coupling between the SI1, the left and right SI1. In Melody, the SI1s are rhythmically active. They're part of the central pattern generator. So the left and right SI1s fire in alternation, and the left swim into neuron 1 is coactive with the left swim into neuron 2. In dendrinotus, the swim into neuron 1s are not rhythmically active in a constant phase relationship to the swim into neuron 2s, and they're not antagonistic to each other. So the swim into neuron 1s are not really part of the central pattern generator for swimming in dendrinotus, even though the behaviors are very similar and probably homologous. If we look further and analyze the dynamics of this, we see that if you hyperpolarize swim into neuron 1, you shut off the motor pattern. So the more hyperpolarizing current you give, the slower it gets until it turns off. Whereas if you depolarize swim into neuron 1, you also turn off the motor pattern. So the more depolarizing current you give, the slower it gets until it turns off. So you get this inverted U-shaped curve. In dendrinotus, if you hyperpolarize swim into neuron 1, you do affect the motor pattern. It slows down. And if you depolarize it, it speeds up. So instead of getting this inverted U-shaped curve, you get this monotonic curve where hyperpolarization slows it down and depolarization speeds it up. So the effects of current injection into these homologous neurons has different effects on the motor pattern. If we look at swim into neuron 2, again, hyperpolarize it, turns it off. Depolarizing swim into neuron 2 also turns it off. In dendrinotus, it's slightly different. Hyperpolarization doesn't really have much of an effect, whereas depolarization slows it down. So again, even though the circuitry looks fairly similar for these two neurons, there's differences in their functional effects on the overall behavior. Now, we've subsequently identified a third cell in the CPG, which we're calling swim into neuron 3 imaginatively. Swim into neuron 3, they're mutually inhibitory across the midline, and it inhibits the other two cells in the central pattern generator. If you hyperpolarize swim into neuron 3, you slow it down, but if you depolarize it, you speed it up. So swim into neuron 3 acts differently than 1 and 2, and you get this monotonic change in the periodicity. In dendrinotus, we also identified a neuron that we think might be homologous to swim into neuron 3 based on anatomical criteria, but its circuitry is really different. It shows electrical coupling, whereas swim into neuron 3 in Melody doesn't. It does still show the mutual inhibition across the midline, but it excites swim into neuron 2 and has no synaptic actions on swim into neuron 1. When you hyperpolarize this swim into neuron 3, you also slow down the motor pattern, but depolarizing it doesn't have much of an effect. So there are differences across the different neurons in the effects that they have on the behavior as judged by the fictive motor pattern. So how did Melody and dendrinotus come to differ in these neural mechanisms? Well, one possibility, for example, is that Melody represents an ancestral state and that dendrinotus has simply lost these synapses. Another possibility is that dendrinotus represents the ancestral state and that Melody has changed. So in this case, if we consider these behaviors homologous, the underlying neural mechanism may have changed. Another possibility is that the common ancestor didn't swim and that Melody and dendrinotus each independently evolved swimming using homologous neurons but putting them together in slightly different ways. We are looking at other species to see if we can sort this out, but it may be just an intractable question. So Melody and dendronotus both swim with left-right flexions. Homologous neurons differ in function, even though the behaviors are probably homologous. The circuits underlying the behaviors differ in their organization, their dynamics, and actually in their pharmacological sensitivity, which I haven't told you about. So homologous behavior can be produced by divergent neural mechanisms. So just to review one more time, different behaviors, looking at Melobi and Tritonia and Tokina, different behaviors, different neural circuits, but homologous multifunctional neurons. Looking at Pleurobranchia and Tritonia, similar behaviors, homologous neurons with similar functions, and neuromodulation correlated with behavior, both within the species and across species. Again, and then looking at Melobi and dendronotus, similar behaviors, homologous neurons, different neural mechanisms underlying the behavior. So, multifunctionality of neurons may facilitate their repurposing in additional behaviors. If you already have neurons that do different functions, it may be easier to change the function of a neuron rather than having to switch a neuron from one function to another. Neuromodulatory actions may uncover the expression of a latent behavior, and the behavior itself is not indicative of neural mechanism. So with that, I want to thank the members of my lab that did this work, Akira Sakurai, who has done just about all the work comparing dendronotus and Melobi, Josh Lilbus, who was studying the modulation, and Jim Newcomb, who studied the DSIs. Thank you.